supercoiled DNA, basically with the help of histones, forms a chromosome. So in humans, we have a total a haploid set contains a total of 23 chromosomes, and a single chromosome makes a single molecule of a DNA. So the DNA is uh, can be divided into can uh, can be seen uh, um, as our nuclear material where genes are located, and in between the genes are the intergenic regions, and there are promoter regions and untranslated or UTR regions. So a DNA is transcribed into DNA, and this DNA further gives a code for the formation of proteins. So in cases wherever there is any sort of change at the DNA level, it will ultimately lead to faulty or abnormal proteins or truncated proteins. Since in human biology and in biological pathways, proteins play an important role, maybe sometimes as enzymes or as an important structure in the cytoskeletal of a cell. So any change at the level of DNA, this can be a single nucleotide change, that is SNV, or indels, which can compromise of a few base pairs, or CNVs, which can constitute CNVs is copy number changes, which can include deletion or duplication of large number of exons of a gene, or maybe an entire gene, or a large number of genes also. So sequencing it is very important for us to know the entire sequence of the DNA. What is the normal sequence of DNA in a healthy individual, and then in cases where we are present, where a disease individual is presented, the sequencing of the DNA of a diseased individual and comparing it to the uh, to a person of who is healthy, uh, it can help us to know that where the change has taken place and how it has it impacted the subsequent pathway. So the Human Genome Project was a landmark global scientific effort with an aim to generate the first human blueprint, because uh, the human genome was still a mystery, and the it was not completely known the how many number of genes are there in the human genome, what is the constitution of the human genome, how much of it is coding, how much if of it is non-coding, and what is the actual role of non-coding DNA, because the coding DNA is the one which will finally get translated into proteins, but what is the initially called the junk DNA or the non-coding DNA is doing in the human genome. So all this it was very important for the scientific community to be able to come up with a human blueprint or a reference human genome to understand the entire human genome, because the human genome contains the code for every biological pathway that is known to humans. So this project, it began in 1990, and it culminated in 2003. So it took a lot of years, because during that time, the only way to sequence the human genome was the traditional method of Sanger sequencing. And in 2023, when this project was completed, it accounted for over 90% of the human genome. So we were at that time able to know the constitution of 90% of our genome. And thus generating the first sequence of the human genome, it provided uh, fundamental information of our human genome, the first human blueprint. And ever since, the study of human biology has improved, and it has advanced. Also, there are newer and latest technologies which help to sequence the genome of not only humans and many other organisms. So this the Sanger sequencing, this was the traditional way that was present during the time when Human Genome Project began, and it is also called chain termination method. This was first developed by scientist Frederick Sanger, and it determined the sequence of the DNA by fragmenting the DNA and then assembling it to generate an entire human genome. So for Sanger sequencing, the it is basically called a chain termination process. In this, the reaction mixture for a Sanger sequencing would contain the template DNA, that is the DNA which has to be sequenced and it is fragmented, and then a primer. This primer will bind to the template DNA, and to this primer then the DNA polymerase would attach the building blocks, that is the DNTPs or deoxynucleotides. A key player here is are the dideoxynucleotides or DDNTPs. On the extreme left, we can see the structure of DNTP and how does it varies from DDNTP. So DDNTP basically, if we see, the hydroxyl group is replaced by hydrogen. So normally what happens during the synthesis of a complementary DNA strand on a single stranded DNA, the once a DNTP gets hybridized, the it can lead to the attachment of the next DNTP. But the structure of DDNTP or dideoxynucleotide is such that it will not allow any further DNTP to bind to it. So DDNTPs, they what they do once they get attached to any fragment of DNA, it will terminate the chain there only. So, and these are also fluorescently labeled. So now in the reaction mixture, what we have is a number of different fragments of different sizes, and they are and where the reaction gets terminated, where the DDNTP is bound. So initially, the gel electrophoresis was used where the in the wells the constituents of the reaction were loaded and then it was run. But in the recent machines, the capillary gel electrophoresis is available. What happens here is that the reaction mixture is injected into the capillary, and the various fragments pass through this capillary. As the fragments pass, the shorter one will move faster, and as they move, the laser is able to detect the DDNTP as it is fluorescently labeled. And as the detection process carries on, it creates a chromatogram. So we can see a set of colorful graphs in the last image. So what the sequencing machine has done here is it has detected the various DDNTPs based on the dye which has been attached to that. So this is how a chromatogram is chromatograph is obtained, and it will give us the sequence of the DNA which was used to determine the sequence of the DNA which was used initially in the reaction mixture. Now, Sanger sequencing is still a gold standard to sequence DNA fragments, but which are of smaller sizes, or sometimes only when we have to validate the result of NGS. But Sanger sequence has a drawback that it cannot, it takes a lot of time and it is not high throughput. So, therefore, there was a need to shift to next generation sequencing, as Sanger sequencing could not meet the need to sequence thousands of millions of people to study genetic diseases. So when the Human Genome Project was completed, it only took the samples of a few uh, individuals and their sequence was determined. But in order to build strong databases and to determine the entire human genome with confidence, it was essential for the scientists to sequence thousands to millions of people who can represent various ethnicities or population groups, and who can form help in and the sequencing of large number of individuals will help in forming or determining the sequence of human genome. Only after that the diseased individuals or the mutations in diseased individuals could be studied. So while Sanger sequencing was the core technology during Human Genome Project, and it made a significant advances because it was the only Sanger only sequencing technique available that time. And during the process of Human Genome Project, the groups that were studying these or sequencing the human genome, they also simultaneously came up with automated sequencing machines. And the first one was Applied Biosystems API 370. This was the first NGS sequencing platform. So after Human Genome Project, the need for newer, faster, and less expensive technologies were felt, and this paved way for NGS or next generation sequencing, and it has largely replaced it for large scale sequencing. So now studying the overview of NGS technology, the basic principle of all the NGS technologies or platforms that are available um, in the market, they involve sequencing millions to billions of DNA or RNA fragments in parallel, and they are high throughput and can help to decode the genetic material. So the key steps in every NGS technology is first to collect the sample. It can be a blood sample or tumor tissue sample, depending on the disease which we have to study or the assay which has to be run in the lab. And DNA or RNA is extracted, which is fragmented, and libraries prepared. After that, basically the library is that the DNA is fragmented, and it is then bound to adapters are small number of nucleotide sequences which bind with the fragmented DNAs, and which help in amplification of these libraries. Then the sequencing is done, and it depends the sequencing for sequencing any type of platform or machine can be used, for example Illumina or Iron Torrent by Thermo Fisher or Oxford Nanopore technology. And after sequencing we finally get data, the sequencing data, but this data is not readily available for analysis. It contains a lot of uh, undesired sequences which need to be trimmed. So the last step, that is in the analysis step, it requires well developed bioinformatic pipelines which help the variant analysis or the variant scientists to be able to read the raw data that is generated after sequencing. So we will study in the subsequent slide how the analysis is done. So this is the basic NGS data analysis pipeline. Sorry. So these pipelines are basically they have been built by our expert bioinformaticians, and it would not be wrong to say that the NGS process is handicapped if we do not have bioinformatics or bioinformaticians or bioinformatic pipelines, which they are continuously developing the more recent and better versions, so that we are able to analyze the tremendous data that is generated after sequencing. For exome sequencing, the data that is generated, it can vary from 5 GBs to 10 GBs. So this type of data, it has to be uh, processed through bioinformatic pipelines so that it becomes available for the analyst. So the data that is generated, the first data which we receive from the sequencing machine is the raw sequence data generation in the form of SRA files. Then the base calling is done. And then during base calling, what we receive is a FASTQ file or unaligned BAM. So during this process, the adapter sequencing, which we just read in the previous slide, which are adapted to the fragmented DNA, those are trimmed because those are not part of the template DNA which we have used for sequencing. So these need to be trimmed. There are other low reads or reads of low quality which need to be trimmed. So this is a way of filtering the sequencing data which we received after sequencing. After that, the whatever sequence we have now in the form of FASTQ file, it has now to be aligned to the reference genome. So the two versions of human reference genome assembly which we have are HG 37 and HG 38. So after we are done with the sequence alignment, we receive SAM or BAM files. So in the extreme right, in the second image, what we can see is this is an IGV browser, or also called integrated genome viewer. So this is an freely available tool, but it is very essential to visualize the reads which we have received after the sequencing. So for this, we have to upload our BAM files into the IGV to be able to see the reads. So this visualization process is also very important because sometimes it helps us to be more confident about the variant or the mutation which we are seeing, and sometimes it also helps us to filter out the false calls. And once we are done with sequence alignment, we have to identify the mutations like SNPs or single nucleotide polymorphisms, indels, insertion deletions, SVs, single nucleotide variants, or copy number variants or CNVs. And after that, we have a list of identified mutations which can be viewed through VCF files. Now, these VCF files can be either further processed to be able to see them in an Excel, or there are various softwares present. Some few are paid, mostly are paid, few are freely available, and we can upload our VCF files, and then we can able to analyze the data. So the next generation sequencing platforms which are available currently and are largely used in research institutions or in clinical labs in diagnostics, these are for example, Illumina. Illumina is a key player when it comes to next generation sequencing platforms, and from time to time it has upgraded its versions. Initially, we had MySeq, HiSeq, and then now we have NovaSeq 6000, and the latest is the X series. Then we have in the in the second row, the second image, it is the Iron Torrent by Thermo Fisher, and the last image which we can see are the portable machines, sequencing machines by Oxford Nanopore Technologies. The size of these machines can vary from our mobile phone to a microwave. They are very portable, and the unique feature of Oxford Nanopore Technology is that it can help generate long read sequences, which we will study in the subsequent slides. So the common NGS platforms, they utilize different methods. They have different principles by which they perform sequencing. For instance, Illumina, the principle used in Illumina platforms is sequencing biosynthesis, and it is the most widely used NGS platforms. So it uses sequences by synthesis technology where DNA fragments are sequentially extended with labeled nucleotides. We'll study this in detail. And then we have Thermo Fisher Iron Torrent, which uses iron semiconductor sequencing. This platform uses semiconductor sequencing, and it detects the pH, the changes in pH when a nucleotide is incorporated into the DNA strand. Then we have Pacific Biosciences, which utilizes single molecular real-time sequencing or SMRT, and Oxford Nanopore sequencing, which uses a platform. This platform uses a nanopore sequencing technique. So the difference between the Illumina and Thermo Fisher and the last two sequencing technologies is that these are able to generate long reads. Okay, So we will now study the technology or the principles uh, which is used by Illumina platforms. And the principles used here is sequencing by synthesis. So the workflow of Illumina sequencing consists of sample preparation, the four major, the basic steps, the sample preparation, the cluster generation, the sequencing, and data analysis. So these four steps are common in across all the platforms, but they have uh, they can be drastically different when it comes to the chemistries they are using. So in this case, uh, for sample preparation, there can be various ways to prepare the samples, but all the methods they add adapters to the DNA fragments. So we can see that the DNA or the RNA that is extracted from the sample, it is fragmented, and then adapters are added to the ends of it. Also, in this case, additional motifs are introduced like sequencing binding sites and regions which are complementary to the flow cell oligos. So, flow cell, in the extreme left, we can see the structure of a flow cell. The flow cell can vary from the version of the Illumina sequencing machine. So, flow cell is a glass side with lanes, and each lane is a channel coated with a lawn of two type of oligos. So this is the flow cell on which actually all the process which are required during sequencing will take place. So the first one is cluster generation. It's a process where each fragment molecule is isothermally amplified, and hybridization is enabled by the first of the two type of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. Then a polymerase, it creates a complement of the hybridized fragment, and the double-stranded molecule is denatured and the original template is washed away. The strands are clonally amplified through bridge amplification. And in this process, the strands they bend over and the adapter region hybridizes to the second type of oligo on the flow cell. So this is how the two type of oligos that are present on the flow cell, these are being uh, utilized during these two steps. Polymerases, they also generate the complementary strand and form a double-stranded bridge. And this bridge is then degenerated, resulting in two single-stranded copies of the molecules that are attached to the flow cell. This process is repeated multiple times and it results in millions of clusters and leads to clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, and the three dash ends are blocked to prevent any sort of unwanted priming. Sequencing begins with the extension. Now, coming to the process of sequencing. So, what happens here is the process of sequencing, it will begin with the extension of the first sequencing primer to produce the first read, which with each cycle we have fluorescently tagged nucleotides which compete for addition to the growing chain. And only one is incorporated based on the sequence of the template, that is based on what is the sequence of the template. According to that, the fluorescently labeled nucleotides will get hybridized, and then so therefore this whole process is called sequencing by synthesis, because sequencing here is done by synthesizing new strands of DNA from the single stranded DNA or template DNA, and the number of cycles they determine the length of the read. So these are the new nucleotides which are being added. They are fluorescently labeled, and the emission wavelength along with signal intensity they determine the base call. When I say base call, I mean what is the base that is present in this particular position. So this is how hundreds of millions of clusters are sequenced in a massively parallel process. So this is also called massively parallel sequencing. And then we have in the sequencing machine the whole entire setup which is able to detect the signals and generate the sequencing data. So this is the principle used by Illumina, that is sequencing by synthesis. The next one we have is ion torrent sequencing, and the technology it uses is ion semiconductor sequencing. So here again, the four major steps are DNA extraction and library preparation. Then we have amplification, sequencing, and data analysis. So till library preparation, the steps will be more or less the same in various technologies, but the amplification and then sequencing, they vary considerably. So in this case, the principle of ion tolerant sequencing is based on detecting protons, that is the H plus ions, which are released when nucleotides are added to the growing DNA strand during DNA synthesis. So as DNA polymerase incorporates a nucleotide into the complement strand, a proton is released, which will lower the pH in the surrounding environment. And this change is then detected by the ion sensor in the semiconductor chip, which is converted into a voltage signal. So after DNA extraction and library preparation, we can see in the first figure that after the isolation of genomic DNA, it is fragmented randomly, and then adapters are added to this fragmented DNA. And we can also read barcode or tag the identification. So what is the barcode? Barcode is a sequence of nucleotides. It is very unique, and it will be attached to, suppose we are uh, performing the sequencing of 5 individuals. We have with us DNA of 5 individuals, because in large setups in big laboratories, individual sample is not uh, sequenced, because there are a huge load of samples. So a large number, it can be 10, 20, 30, 40, they have to be sequenced in a single day. So to perform the sequencing, a barcode, that is a set of unique nucleotides with a unique sequence, is attached to this fragmented DNA. So after we have attached these barcodes, we can pool this DNA. So this pooled DNA will help in making a library and then performing the sequencing. And after we are done with the sequencing, we can demultiplex. That is, we can segregate the different sequencing results which we have got based on the barcode which we had attached to the individual sample when we begin the library preparation. So in during amplification, once we are done with library preparation, the DNA fragments, they are bound to the surface of tiny beads. These are the magnetic beads, and these are also called the ion sphere particles, or ISPs, and they are amplified using a particular type of PCR reaction called emulsion PCR. So after the emulsion PCR reaction is done, the beads containing the successfully amplified DNA, they are enriched by centrifugation, and only those beads which contain uh, the amplified DNA, they are then subjected to sequencing. So the amplified beads are then loaded into microwells on the semiconductor chip. So the way we studied flow cell in case of Illumina, here the base, the platform where the all this reaction and sequencing will take place is a semiconductor chip, and each well on the semiconductor chip, it will hold a single bead. So these have a very elaborate, these flow cells or semiconductor chips, they have a very elaborate detailed structure which is not cannot be seen through the naked eye, and we cannot directly load a sample by a pipette onto a flow cell. So for loading a sample, we again have dedicated machines which where we put all the reagents, the flow cell and everything, and it loads the sample and all the reagents onto uh, the flow cells or semiconductors based on the sequencing machine we are using. So in this case, the amplified beads are then loaded into microwells, and a single bead will be present in a single well. So during sequencing, nucleotides are sequentially flooded over the chip, and the incorporation of the nucleotide is detected by the ion sensor, which is present in the chip itself. So we can see here in the diagram that uh, if a nucleotide that is matching the DNA template is releases the hydrogen ions, it changes the pH of the solution. And the ion sensor on the chip, it will detect the change in the pH as an electrical signal. So through detecting these signals, the nucleotides that are incorporated during the sequencing process, the sequence of those are then determined. And of course, the data analysis is done after the sequencing is done. So we see, if we compare this to the Illumina synthesis, sequencing biosynthesis, we see that the four main steps, that is the sample preparation, library preparation, amplification, sequencing, and data analysis, the steps are the same, the sequence is the same, but the basic chemistry is quite different. So the reads which we obtained through these two technologies, which we have studied so far, these are the short reads. And now the most recent technology which is now being introduced into the sequencing market is long read sequencing, and one of this is provided by Oxford Nanopore sequencing technology. So they have developed third generation sequences that are portable. So these are so handy and so easy to use that sometimes when a scientist or an investigator wants to study flora or fauna of of a particular place, the investigator chooses, he can take that this sequencing machine along with himself, and it can he just only has to attach the this sequencing machine, it is portable, to the laptop, and then the sequencing will be performed, and the data that is being generated in during sequencing can be studied in real time by the investigator. So in 2015, the first Oxford Nanopore technology, or ONT, sequencing device was released, and it had an entirely new way of sequencing DNA, because it here there is no need of DNA polymerase. Instead, it's based on a small barrel-shaped protein in a membrane called a nanopore. So in this figure, we can see that in a in a membrane is embedded a nanopore, and it also contains a motor protein which is yellow in color on top of it. Which is and whenever the actual sequencing, the sequence of the template DNA is determined by the change in electric current, which takes place when this single stranded DNA, template DNA, flows through the nanopore. As the DNA travels through the nanopore, it will obstruct the current flowing across the membrane. So we can see here a double membrane which has trans and cis sides, and there is difference in the flow of difference in the current on the two surfaces. So as the DNA will travel through this nanopore, it will obstruct the current, and all the four bases of DNA, that is A, T, C, and G, they are of different size and shapes. So they will cause variations in the current. And these variations, basically, these are measured by an electronic chip. And then we have an elaborate algorithm which will convert the DNA into a sequence which can be read. So here we can, on the right side, we can see a graph against current and the time, which can actually help in determining the sequence. This process is also, the determining the sequence is also called base calling. So what we see here, there is no need of a polymerase and only a nanoprotein that is embedded into the membrane. Into this is an artificial membrane, uh, and this is the motor protein. It helps to unzip the DNA helix. This is the DNA which we need to sequence, and this is how the changes in the or the variations in the current flow are studied, and the base calling is performed. So these are the major sequencing technologies which are available. They have their unique way of sequencing, and they have their own application in various fields. If we compare all these DNA sequencing technologies, the first one was the Sanger, which is also called the first generation. The method here is also the synthesis, but the throughput is low, as the and the error rate, but the error rate is less. So when we talk of next generation sequences, the Illumina, it what it gives is it is short reads, and the technology or the principle it uses is synthesis. Throughput is high, and the read length is 100 to 300 base pairs, and but with a slight increase in error rate. So sometimes these next generation sequencing, as the sequencing technology advances, there is chances of increase in error rates. So sometimes it there is complexity in the data which we receive from next generation sequencing, and which has to be sometimes confirmed with Sanger sequencing. So that is why I said that Sanger is still a gold standard. So the capillary-based nanopores or single molecule real-time SMR technologies, what they are now providing us is long read sequences. So long read sequences, they help in understanding the variations that take place at a larger stretch of DNA. For example, CNVs. There are few diseases in which CNVs have a more important role to play rather than a single nucleotide. So if we talk of the advantages of NGS, of course, it is high throughput because it can sequence millions to billions of DNA fragments simultaneously and can produce a lot of data in a short period. So, and it is also cost effective. When this all sequencing thing on technology developed, the sequencing costs were really high. But as the technology has advanced, there has been more emphasis on how to reduce the cost, because ultimately when we talk of the diagnostics, the main focus is to reduce the cost so that people of various socioeconomic backgrounds, they are able to have access to this recent technology, because in few diseases NGS is only able to provide answers. Also the precision and accuracy, it is also increased by the recent bioinformatic pipeline that are coming up, and there are more advanced versions now which help to increase the accuracy and precision. And not only human or human genomics is benefited by NGS, but it can also help in microbiology or studying the evolutionary biology. So the capabilities of NGS are that it is a high throughput sequencing, and with time, the latest technology in NGS has helped in sequencing millions to billions of DNA, and therefore now we have uh, genomes sequenced of many organisms now. Also, the whole genome sequencing, sometimes exome sequencing, that is sequencing all the exons of few genes, or the entire human genome is not able to provide answer. So sometimes there is need for sequencing the entire genome. So this has now this is also now accomplished by NGS. And not only DNA, but NGS can also be used to analyze the transcriptome, that is the complete set of RNA transcripts in a cell. This is particularly essential in studying the gene expression, and especially in case of cancer or somatic cases. So we have targeted sequencing, which we need not sequence the entire genome. When suppose a doctor is very sure that the patient is affected with seizures or this particular kidney disease, then a set of genes can be sequenced. Those genes which are implicated in the disease with which the patient has been presented to the referring clinician. And also in metagenomics, so NGS can sequence the data of complex microbial communities like the gut microbiome. So the gut microbiome is everywhere now because it has been linked to a lot of diseases. So the sequencing of these microbial communities is now providing answers to a lot of health conditions. And epigenetics, where it can be used to study modifications on DNA, such as DNA methylation or histone modification, which helps in understanding gene regulation and cellular processes. So if we look at the application of NGS in a more detail, we see that it has a broad range of applications across various fields like genomics, diagnostics, and research. If we talk of clinical diagnostics, NGS is able to provide answer to many inherited genetic disorders like rare and undiagnosed diseases. And there are still few communities or remote communities which are affected by some kind of genetic disease, but they are not aware of it. And if there is some particular research group working on it, they can decipher the exact mutation which is affecting the entire community. Also, in case of cancer genomics, the somatic mutations, the acquired mutations, the gene fusion and copy number variations, they are largely seen in tumors. For example, BRCA1, the copy number variation in BRCA1 is a predominant mutation in breast cancer or ovarian cancer patients. And also in prenatal testing, for example, in case of NIPT or non-invasive prenatal testing, where there is no need to go for invasive tests like amniocentesis, um, where there is need to remove a little amount of amniotic fluid, but it can also harm the fetus. So the non-invasive technique which we have now is NIPT, in which the circulating DNA of the fetal, which passes through the placenta and is found in mother's blood, it can be extracted and then it can be sequenced. So prenatally, the expecting mothers, they can be informed of the possible risks that their fetus is carrying, and therefore the reproductive decisions can be made based on the NIPT test. Then we have personalized medicine. For example, um, uh, in few individuals, they have their they can be good drug metabolizers, or there are few individuals. um, so basically, there are various genetic variations which can affect the drug metabolism, and it can vary from individual to individual. So understanding the genetic variations can ultimately lead to understanding the effect of drug that can the effect of drug in a particular individual, and help in uh, customizing a treatment plan for an individual based on the person's genetic makeup. In cancer treatment also, the various type of targeted therapies which are designed, they are based on the type of mutation a cancer patient is carrying, because a few type of a few type of genetic mutations, they render a person unfit or render a person resistant to a few kind of treatments. So in those cases, by determining the sequence or any mutation in the cancer causing gene, can help a clinician decide if there is a particular type of treatment will be helpful to the patient, and if not, the patient will not be subjected uh, to a treatment which is not affected to the patient. And also in rare disease treatment, which we have already just discussed, that there are few genetic mutations which can help in understanding rare and undiagnosed diseases. So as we are studying the applications of DNA of NGS, so for a beginner, for a person who have just stepped into understanding NGS, it is not that the entire genome has to be sequenced to study the variations. Based on the disease of an individual, there are panels that have been customized. For example, there is a person who has come with there's a particular type of kidney disease called Alport syndrome, and there are only 4 to 5 genes that are responsible for that syndrome. So in that case, the diagnostic laboratory will only sequence those 4 or 5 genes. So what it will do, it will cut the cost of sequencing, and it will benefit the patient, that the patient has to pay a very less amount for the test, because the biopsy, the renal biopsy report is um, very strongly suggestive of Alport syndrome. And when such a patient comes to laboratory, the scientists in the laboratory, they already know that okay, for this person we have 5 set of genes which are established in Alport syndrome, and we will only target these. So this kind of treatment or this kind of sequencing is called targeted gene panels. We have targeted gene panels for specific diseases. Then we have exome sequencing, where all the exons, when I say exons, means exons and not the introns. All the exons will be sequenced, and these can help in a lot of cases. So sequencing the exome of an individual can provide a molecular diagnosis to the patient. And only a few cases, there is a need for whole genome sequencing. Again, coming back to the applications of NGS, it is also widely used in microgenomics to understand the metagenomics and sequencing microbial communities directly from the environmental samples like soil, water, or gut microbiome, and pathogen detection. For example, characterization of bacterial, viral, and fungal pathogens. So whenever we have outbreaks and that virus or pathogen has not been known to the scientists, so NGS or sequencing of these pathogens can help in characterizing those pathogens. And then in antimicrobial resistance also, because it can help in tracking the spread of antimicrobial resistance by identifying the resistant genes that are present in the microbial populations. Then extending the applications of NGS to agriculture and environmental applications, it can be uh, used to sequence genes which are responsible for a few desirable traits in plants, and then helping crop breeding programs in animal breedings, also in selecting a few traits in livestock and selective breeding programs, and then also in environmental monitoring of environmental changes through the analysis of genetic material found in water, soil, or air. Then also in forensic science, uh, we know this term DNA fingerprinting or DNA profiling. So in cases where there is in scenarios where on or crime scenes, we find the blood sample or semen sample or hair sample of the criminal, and it can help um, uh, the investigators to understand or to know who the actual culprit is. Also, sometimes confirming the paternity in those cases. Also, DNA fingerprinting or profiling can be used. So, our human genome is very diverse, and there are few complex regions, there are repeat regions, there are short tandem repeats, and the for example, short tandem repeats, the number is very specific for an individual. So the sequencing technologies and new assays that are coming up, they utilize the diversity of the human genome and what is the best assay that can determine the type of change in the human genome based on the complexity of the region in which that change is present. Then also in cancer research, um, um, sequencing can help to understand the mutations which actually helped in driving the normal tumor or a benign tumor to a metastatic cancer, and also in liquid biopsies where the analysis of circulating tumor DNA or RNA from the blood samples can be the circulating tumor DNA can be isolated, and this is also again a less invasive technique because to analyze the DNA which is obtained from the tumor tissue of the patient can sometimes be not suitable to some terminally ill patients. And lastly, to know the challenges which we are facing with NGS is that since it produces massive amounts of data in GBs, for a single individual, it can reach up to 10 GBs. So we need powerful computation tools as well as proper data storage methods. Then the error rate in different NGS platforms um, uh, can the error rates can be high, so it is important to choose the right type of technology for the right type of test. Also the team, the people who are analyzing the data, they should be expert in their fields, and they should know what are they analyzing and what is the complexity that can be associated uh, with the analysis, and so that they are very cautious at every step of analysis. So, and again, the cost of analysis is very still high for many people. and it has decreased over time. And again, this is still a challenge for many people, the cost of NGS. But still, I think in coming years it will reduce further, so that uh, a common person can have access to NGS. And again, the complexity in data interpretation. So, sorry. So, for example, the referring clinicians or the doctors who are treating patients with a genetic disease, they should be aware that with this kind of disease, a particular test will be beneficial. For example, if I talk of Huntington disease, there is a there's a trinucleotide expansion in HTT gene. So NGS cannot detect that. For that, we have fragment-based PCR. So based on the complexity uh, of the human genome and the type of disease, it is very important to go for the right kind of assay. And the emerging trends which we are seeing in NGS is that now it is also allowing for single cell sequencing, and it enables a study of cellular heterogeneity, very important, especially in cancers and tumors. And then we have long read sequencing. There are more and more technologies which are coming up, providing for long read sequencing, which will be useful for assembling complex genomes and studying structural variations. Then also direct the RNA sequencing, because many technologies, what they do, they extract RNA sample, then they convert the RNA to cDNA, and then they do the sequencing. But the cost and the time will be reduced if direct RNA sequencing is done. And then we have real time sequencing. As I mentioned in one of the previous slides, that these are portable machines which can be carried. On-site sequencing can be done in remote locations, and real time, and the data can be analyzed in real time. And with this, I end my presentation. I know a lot of it could be added, but keeping the time constraint, I added the main concepts of NGS. I hope this will be very helpful to you, and please um, feel free for